We are XPose Therapeutics based in San Francisco. This is the reason we are called XPose Therapeutics is because we try to do drug discovery starting with poses of molecules bound to crystal structures of proteins. So it's really starting with X-ray poses. And the other reason is you can see the X-ray diffraction pattern here. It's a simulation of an X-ray diffraction pattern. So we are trying to expose how small molecules bind to protein targets. So there's a reason behind the name. We're trying to reimagine the drug discovery paradigm as we think, initially looking at targets that are involved in DNA repair. So you can see a broken strand of DNA here, looking at endonucleases and so on. So the key focus of what we do and our approach is looking at rapid experimental hit generation and then hit to lead generation for drug discovery on structurally enabled targets. These are targets that are that already have either scientific validation or clinical validation, and in many cases, at least in the ones that I'm going to show you, these targets can be crystallized, but nobody has crystal structures of small molecules bound to these targets yet. So there's no experimental evidence of how ligands interact with the binding sites and so on. And what I'm going to describe to you is a way we do screening, where we in fact screen compound libraries directly by X-ray crystallography. When I started, when I was an undergrad, I started doing biology and biophysics and protein folding. I spent a lot of time on learning molecular dynamics simulations and protein folding and so on, protein structure prediction. These were the early days of doing these, but it was computational methods and computing power was not as good as it is now, and even now. And so I moved to X-ray crystallography, and over the years, of course, over the last 20 years, there have been massive developments in computing and algorithms and so on. But at the end of the day, you still need experimental structures and empirical validation of binding. And so we have decided to follow an approach where we start with looking at experimental data to do any drug discovery. I know a question just came up at the beginning about easy to crystallize targets. So this is a misnomer. Just because things are structurally enabled doesn't make them necessarily easy to crystallize. You can, in fact, have lots of complications with crystallography, even for crystals that can be readily generated. So we can discuss some of these in Q&A later on. But structural enablement doesn't mean it's easy, number one. And the second thing is, it's also incorrect to think that everything that has been crystallized is only because they're easy to crystallize. That's also incorrect. Most of it has taken years and years of work to do if you follow what's going on in the PDB, other than very simple targets. Please feel free if anybody wants to. I think you're muted, but I can try to take some questions as we go along, or we can wait till about 20-25 minutes into the talk when I'll hopefully finish and we can go into a Q&A there. So we have a unique platform that identifies and converts novel binding sites and binders into active compounds for use as both small molecule inhibitors and targeted protein degradation ligands. We call this platform SaXSpeed. It's actually a linear algebra term. I won't go into the math, what it means in math, but for us it stands for SAR by X-ray poses quickly. Yeah? So we look at everything as X-ray poses from crystal structures, and then we try to do rapid structure-activity relationship development of compounds based on that. So again, a key feature is that it's both a need to identify novel binding sites and novel binders, and then convert them into active compounds. Yeah, so these are the two important things to realize here. And while we are on it, I should explain what we mean by identification. So as you know, a lot of drug discovery happens by library screening at the beginning, right? It can be high-throughput screening with biochemical assays, HTS. It can be computational screening. It can be biophysical screening like surface plasmon resonance or nuclear magnetic resonance. However, none of these methods tell you where the binding site is really, other than computational screening, where you start with a known binding site. But if you just have a protein structure or a protein molecule, none of the other methods will tell you where the binding sites are, unless you're using a biochemical assay that is optimized for a functional site. If you have a new site that you want to identify, you're not going to be able to find it. And biophysical screening can identify binders, but it cannot tell you where they are. So the only way to really do that is by crystal structures. And so in our process, we are able to get both of those at the same time simultaneously. We're able to find sites and then look at what binders bind to them. This is a graphical illustration of our X-shaped SAR by X-ray poses quickly platform. So the first prong of the platform is to use high-throughput X-ray crystallography for novel binding site hit identification and site identification. The second part is to quickly convert that into chemical evolution. That's where a lot of ComChem goes in, which is why I think it's great to have an audience that is at least looks like 60%, 40% of it is medchem and another 20% ComChem or so, 60% chemistry. So that's a pretty important thing that we would talk about today, and the value for using structural data to drive that. And then the last two prongs are how do we use that information, right? So the good thing is that we can use them simultaneously without any depletion of focus. We can use them as small molecule inhibitors. But when we have novel binding sites, we can actually use them as hooks for targeted protein degradation ligands as well in a parallel approach. So that really gives a dual power to the platform for doing this. It's a fully developed, validated, and ultra-high throughput platform. The first part of the strategy is the library screening. This is done by my other company, Accelero Biostructures, that really focuses on, it's a crystallography company that focuses on all things protein X-ray crystallography and structural biology. XPose partners with Accelero for using access to that platform in the first part of it. And then once the compounds are identified, once the screening is done, XPose's specialty is then trying to take them on into chemistry and biology, and then look at therapeutic indications that it can be applied to, and so on. So the therapeutics part of it comes from XPose really, and then Accelero just provides the foundational screening approach to it. So the way we do it is, what Accelero does and what XPose capitalizes on is marrying of two very well-established methods in drug discovery. One is fragment-based drug discovery that has been around for quite some time now, over 20 years, and with about 5 or 6 molecules in the clinic already. And then X-ray crystallography, of course, which is also very well known. I mean, those of you who are computational chemists, I'm sure you spend a lot of time looking at crystal structures from the protein data bank and so on. So the two very established methods. What was missing was the gap, a big gap really. It sounds like a small gap, but it's actually a very big gap of combining the two approaches. So the conventional way in which things are done in drug discovery is that you use biophysical or biochemical screening or computational screening to identify compounds from library screening. Then you triage them into a smaller set that is then pushed into crystallography. So our plan was to turn the paradigm on its head. Why don't we just start with screening right from the beginning with crystallography, right? So although this sounds conceptually to be straightforward, it's not really straightforward because you have to generate hundreds and hundreds of very, very reproducible crystals that can diffract very well. So there's a big intense focus on crystallization at that point. And again, this comes to the point of addressing what we mean by easy to crystallize, right? So even if something looks like it's easy to crystallize, it's not necessarily optimized for library screening. So to generate hundreds and hundreds of these crystals that have to be very reproducible, of high quality, then there's a lot of effort that goes on into compound management to be able to expose them to hundreds and hundreds of compounds and then try to do that process. And then finally, once those are obtained, then you have to collect data on hundreds of crystals at a time, collect hundreds of data sets, process them, do structure determination on them, analyze their structures, look for ligand density, and then of course try to come up with binding sites and binders, as you can see in these pictured and colored compounds at different sites of the protein, right? So the value of this approach is that we can quickly identify different binding sites that exist on the protein, yeah, and then move them into biology and chemistry very quickly. So once you know where the binding sites are for biology, to be able to evaluate the impact of that on the functional on functional biology becomes very significantly enabled. And then for chemistry, once you have binding sites and starting compounds, it's easy to start looking at IP landscape and how that might evolve you can look at synthetic tractability, and then ComChem can use those poses as anchor points to then do hit expansion around them, right, to be able to grow those compounds and fragments out as Biosolvit's software actually enables to do in a fairly robust manner as well. Once you have that, of course, then we can cycle that back into X-ray crystallography and move on from there. So again, we can use these as enzyme inhibitors or modulators, as well as compounds for targeted protein degradation warheads, right? And in our case, in case of DNA damage response that we're looking at in the beginning, we can use these compounds in a therapeutic modality, either in combinatorial treatment or in synthetic lethality. What do we get from this approach? So we're really trying to visualize success early on in the process by a structure-first approach, right? So if you look at this magic quadrant on the X, and we look at X-axis as having, as representing 3D information, and then the Y-axis as IP value, we span an entire spectrum in the drug discovery field, right? So there are projects that have no experimental 3D information available. So they're pretty much driven by biophysical assays and biochemical assays and computational screens. And then on the right side of the spectrum, there are projects that have experimental 3D structures, and we look at if we can actually enable that with fragments in primary screening or hit generation and iterative structure-guided optimization. And on the Y-axis, you can have either very little IP value because you don't really have that information on what's going on in terms of protein-ligand interactions, or you can have pretty high value. So there's a lot of projects that span the spectrum of this. We think we are right at, on the top right-hand corner of it. So really high IP value starting from looking at experimental 3D structures right from the beginning. Yeah. We did publish 2 papers last year on, first was an approach to our fragment and structure-based drug discovery for developing therapeutic agents targeting DNA damage response. And then we published another second paper where we're looking at a specific target, DNA polymerase data. So I'm just putting them up here because it gives you something to go back to and look at the context of why we're doing what we're doing and how we're actually being able to build the technology based on our history of developing high-throughput crystallography before that for the last 20 years. So DNA damage response, this is known for a long time now that there are many cancers that arise from inherited or sporadic defects in DDR. And this provides a unique targeting opportunity. And these defects can occur from endogenous factors like replication stress and cell metabolism, oxygen radicals within the body, or they can be exogenous factors like radiation, viral infection, chemotherapy, and so on. A lot of it, of course, comes from sun damage on a day-to-day basis, which is why we put on sunscreen, right? But that's UV radiation. Now, there are various mechanisms by which DNA repair is controlled in the body. There's mismatch repair, nucleotide excision repair, base excision repair, translesion synthesis, homologous recombination, non-homologous end joining. And if on the right panel, you see this graphical representation of these different pathways. And there are many, many players in each of these pathways, right? So there's a lot of work that we can do in DDR in terms of modulating DNA damage response and in terms of looking at novel oncology targets. However, the field really started growing in terms of drug discovery, other than the basic biology that had been done for the last many, many decades. Drug discovery really took off in the last 5 or 6 years after the Nobel Prize in 2015 on DNA damage repair. And since then, there are 10, 15 companies or more looking at how to develop novel therapeutics against DDR targets. However, most of them really focus on well-known targets like PARP and ATM and ATR kinases and so on. However, as you can see, there's so much that can be done, and many of these are structurally enabled. So a lot of our interest has been in looking at novel targets that we can push into and really enable drug discovery on. And this is a chicken and egg problem. So unless you have hits to start with, you cannot really look at validating them, right? So how do you have hit generation that you can do that can give you high value hits that you can start testing in assays? And so that's some of the philosophy that drives us to be able to generate protein ligand structures very quickly that we can then think that these are some things that we can move along and look at drug discovery from from a therapeutic standpoint, from a industry standpoint, not just do screening and get some compounds that don't look good and don't know where they're bound and publish them. That's not our interest. Our lead target is APE1. It's an apyrimidinic endonuclease. It's involved in really chopping up DNA backbone once they're damaged. This is a graphical representation of that. But this is a target that is a single target, but with multiple clinical deployment strategies that we are looking at. So it's in fact implicated in temozolomide and glioblastoma based therapies for glioblastoma. We can look at it in, and this is really a combination strategy to look at combining APE1 inhibitors with these therapies that are already in the clinic, right? So there are antimetabolites that can be used for gastric cancers or cisplatin for lung cancer. And we have already some evidence that when we use our novel APE1 inhibitors, we can potentiate the effect of these other DNA alkylators that go into conventional therapies. And we are also looking at synthetic lethality or synergistic landscape, where we're looking at combining APE1 inhibitors with PARP inhibitors, DNA protein kinase inhibitors, ATM inhibitors, and so on. I'm going to skip through some of this. This is really background work on the value of this target, but it's really known that if you knock out APE1 in cancer cells, you get cell killing and arrest. But in normal cells, if you knock it out, you just get mild phenotype. And we know that cancer cells are rapidly dividing cells. So there's evidence that if you just knock it off in cancer cells, we can really make an impact on looking at the therapeutic for APE1. There's also very other interesting evidences and reports that have come out. There is a case of, this is a nice paper to go back to look at when you have the time. This is a 10-year study that came by the NCI, National Cancer Institute here in the US, that was published last year, and they found some things, some patients that are exceptional responders. So they have in a treatment for glioblastoma with temozolomide, and the glioblastoma is one of the most difficult cancers to treat with a survival rate that is very low. But they found that this patient, when they were treated with temozolomide, they had an exceptional response that lasted over 10 years, and they found that there was an APE1 mutation on it, suggesting that if you can target APE1 inhibition along with temozolomide treatment, you can get a good value for glioblastoma treatment. And these are some of the papers that discuss looking at synthetic lethality in combination with PARP inhibitors and DNA protein kinase inhibition and so on. So what are the results that look like? So these are crystals that we of APE1. Again, it's a very different ball game when you're solving a crystal structure to get one structure. It's a totally different ball game when you are looking at generating crystals for screening hundreds of compounds in a screen. But what we did with this is, once we had optimized and reproduced those conditions and scaled up crystallization and screened the library, we were able to get about 25 crystal structures and two binding sites. So this is totally novel, yeah. So these are first crystal structures of APE1 bound to ligands, despite the fact that this target has been studied for over 15 years. There are many papers on it. People have tried high-throughput screening, computational screening, pharmacophore modeling, and so on, and there is no experimental data so before our structures on how and where these molecules are binding or could be binding. And in addition, we identified a novel site as well. So there are three take-home messages here. One is once with the screen, we can get a number of crystal structures that can actually parallelly be considered for medchem optimization. Yeah? So that's a pretty powerful strategy to begin with when you have like 20 compounds to look at and the structures at the same time, as well as potential allosteric sites from novel binding sites. So this really jump-started the program at XPose Therapeutics on this target. And the other question that comes up sometimes in crystallographic screening is about hit rate. Again, that depends on the target really, and binding sites and how the target shape is and if there are binding sites available or not and so on. But we can address that in the Q&A. So a few more slides and I'm going to stop after that. An important question when we first started doing this was, okay, so you get structures, yeah, so what does it mean? Are they active? What can you do with them? And that's where the power of the Saxspeed platform and following them up into lead generation comes in, right? So we were actually able to do a hit expansion of this, one of the original hits out of 20, by the way. So that again is limited by chemistry and biology throughput, right? So we took one of those hits that we deemed to be important based on its chemistry and binding site and so on. We then expanded that out to 400 compounds. And within a few weeks, once we had the assays set up to do test the compounds in assays, we can cycle this through fairly quickly. That's why we call it as SAR by X-ray poses quickly. So we were able to go from an initial fragment hit that was less than 2 millimolar IC50 into a compound that was 500 nanomolar IC50, and then further optimize that to about 250 to 300 nanomolars now, all within a matter of a very short time, a matter of a few weeks or a couple of months, depending on if you don't count just hit expansion and chemistry and assays, but just the ability to quickly move on from something that is low potency to a high potency. So this is really a huge gain, right? From 2000 micromolar to 0.3 micromolar. So we then not only were we able to do that, of course, we tested that in the biochemical assay, but we also showed that it has specificity over other APE endonucleases. We showed that. Then we were also able to quickly move into cell biology assays. It's a cell killing assay. And we showed a couple of those. I'm going to skip over these as well, but we essentially show that we can potentiate the effect of DNA alkylators and chemotoxins by using our APE1 inhibitor as well. And just to drive home the point of the value of this, it's not a one-trick pony kind of thing. It's really a very sustainable and robust approach that we can use for multiple targets. This is another DNA repair protein. This is a polymerase now. We ran this screen on a protein DNA complex. This is what its crystal structure looks like. But this is an example. In this case, we obtained about 7 hits. Again, identified 2 totally novel binding sites. This is an example of a partial depiction of a hit that we obtained. We published this, so I'm showing it here. This is not the full fragment hit, by the way. So this is only part of it because we couldn't disclose the entire chemistry on the binding site. But suffice it to say, it just shows what the quality of electron density and maps are, right? You can really look at how nice this interaction is between the hit and one of the side chain residues. So we're looking at this target in terms of cisplatin resistance in ovarian cancers and a couple of other applications. But we, again, of course, solved this by library screening. And we were able to quickly move. In this case, we haven't expanded the compound set a lot yet. We just moved from the initial hit, one of the initial hits, to a 40 compound expansion. And we already got about a 10 times improved potency starting from there. So I'm going to stop there. I think it's about 30 minutes, and we can certainly move into some discussions.